Okay, thank you for that introduction. And it's nice to be here again in Zurich. It has been a while since I was here, but it's always nice to come back to Switzerland. So as you can see, my model organism is Daphnia, but I will not only tell about Daphnia, but the biggest part will still be the Daphnia. And especially the good thing is with Daphnia, they are very phenotypically plastic. And as you will see, the microbiome plays an important role in that. So I will try to stress that. But before I go on, I want to put it a little bit in a bigger picture, and as you all know is and as we know is that actually there is a lot of stress on aquatic organisms and organisms in general, but especially in aquatic organisms, we see that you have all this kind of environmental stressors, but on the other hand, they also have important symbioses with particular microbiota that helps them to deal with these different stresses. And I hope I can convince you today that these microbiota do play a role for this host to deal with these multiple stressors. So actually, there are two different points I want to give you through my talk. And the first is actually the role of the microbiome in eco-evo dynamics, so eco-evolutionary dynamics, which are typically characterized by ecological interactions and evolutionary interactions happening at the same time and also interacting with each other. And one point that I want to focus you on is the role of the microbiome in this eco-evo dynamics and a way how we see that, and that's then the second point, is actually the interaction of host genotypes with the microbiomes to deal with this particular environmental stresses that I said. So a crucial aspect is the host genotype microbiome environmental interaction, and we looked at for different multiple stressors. So the one that I will talk most about is the cyanobacterial stress that you can see here. So these are toxic cyanobacteria, which are actually also bacteria, but Daphnia is exposed to them, and you will see they respond via different ways. And we also look to a few other stresses as well, but I will mainly focus on that one. But before we go to the aquatic systems, I will put it a bit broader and then indicating the role that microbiota play for hosts and what the effect of that is on the larger ecosystem. So we actually know that there are a lot of factors that are influencing these microbial communities in your host. So we know that the host immunity can have an effect and is structuring the microbial community. But also, like I said, these environmental effects are also directly influencing this microbial community. And next to that, you also have the species interactions of your microbial community. So actually structuring all kind of eco-evo interactions in the host and the host microbiome. But as I said, so we see that on different levels. So first of all, we have this eco-evo dynamics on the microbial community. So you have evolutionary responses and ecological responses really at the microbial community. But very important, what we see is that these interactions also modify your host phenotype. And it's by modifying your host phenotype that you can actually have an effect mediated by your microbiome on the wider environment. So the host is only a part of a bigger community in your ecosystem, and that's actually one of the things we want to point. So you have these phenotypic modifications which structure the interactions of your host with the wider community, and that's actually what we call the bottom-up hierarchical feedback of the microbiome to the host community. And opposite, of course, we also have the top-down hierarchical feedback. So it's actually a loop of feedbacks that are structuring each other, and that the crucial aspect in that is that your host phenotype is modified by that phenotype. And given that that is really important, this modified phenotype, we see that as an important point in this hierarchical dynamics. So these are typical, the different levels that you have of the eco-evo dynamics at your microbial community. So evolutionary responses, ecological responses, and that this modifies your microbial phenotype. But on the other hand, we also see that the host phenotypes plays a particular role in this whole community of the wider environment. So, and I hope that I convince you today by actually showing you that Daphnia is one of the systems that is really reflecting that. And then why Daphnia? Well, most of you know the system, but so they are suspension feeders, they are keystones. So that means that if the host phenotype is changing, you find a strong effect on different levels of your ecosystem. So it's an important player. It adapts relatively fast. It has a diverse microbiome. We know that by now. And of course, it has the both modes of reproduction. But very important is the phenotypic responses. And probably the best known response in here is the helmet formation that they typically do for fish predation. So that is a famous phenotypic response that we see. But also towards other stresses like the cyanobacteria, we also see that your phenotype can modify. And I will show you that with a few experiments that we did. But before we could actually study this modified phenotypes, one of the crucial things that we needed to be able to handle in the lab is creating axenic lines. So axenic Daphnia lines, because by doing that, and that's what you see in the top panel, so what we actually do is take the parthenogenetic eggs out of the Daphnia, and then we let them hatch. We expose them first to disinfectants, and then we let them hatch at a crucial phase in the development. And then what we additionally do is take guts from other Daphnia, squash the guts, and then do transplant experiments. So by the fact that we can do that, we can actually control the system relatively well and see what effect the microbiome has on the phenotype of our host organism. And additionally to that, so we also sequence the bacterial community, both from community perspective and then also from functional genomics perspective to see what particular role the host microbiome plays in particular responses, especially with respect to detoxification and dealing with multiple stressors in your environment. So first of all, what we saw is, most importantly, what Martijn saw is actually that if you here compare, so if you compare germ-free Daphnia, so axenic Daphnia, with individuals that had microbiomes, you clearly see that having a microbiome in the symbiotic treatment has a positive effect on Daphnia performance. That's something we systematically find. So they need the microbiota to be able to develop and to reproduce. So that's first of all very important. And then secondly, what we also see is that if you compare the microbial community of the gut versus the whole organism, that there's a clear distinction between those two different communities. So that already shows that even in such a simple organism as Daphnia, the gut microbiome community is somehow probably structured via different factors. And then the question is, what is structuring this community? So there can be different factors that do that. And so the question is, what pool of microbiota do you actually get in your gut, depending on what is in your environment and what plays a role? And as one of the hypotheses we have is that the host genetic background, so which genotype you have and the environmental condition is actually both important in structuring that community. So that's the first effect that we hypothesized. And so Martijn did experiments in here. So what you see on the vertical axis are 5 different clones. So we used 5 different genotypes so we can keep them asexually in the lab. And then we expose them to 4 different microbial inocula on the horizontal axis. And then the question is, what is the relative impact of the bacterial plankton? So from the inoculum, the bacterial plankton is the inoculum that you use. So there was a bacterial plankton coming from 4 different environments. So one environment was a Daphnia jar, and the other 3 environments were more natural environments. And in what way does the host genotype has an effect on that? So if you look to the sequencing results of the microbial community of the gut, so the results that you see in here are actually the sequencing of the gut microbiomes, and you see, first of all, which inoculum that they got was actually also affecting the gut microbial community, because you see on the right in here, there was a Daphnia jar, so it's called hay extract, so it was a Daphnia jar with hay, so you clearly saw that that community was clearly different from the other communities. And then when you look more in detail what particular strains are present, that clearly see all the yellow strains were this particular inoculum. So what you give to the Daphnia is also what in the end you get in the Daphnia. So the microbial inoculum really has an important effect on what you finally see in the gut. But very interestingly, and it was even more interesting to me, is that, so on the bottom here, you see the 5 different clones, so the 5 different genotypes that were made axenic or sterile. Then you see that the different strains that they have, the community that they have, is also different. So depending on which genotype you're looking at, they had a different microbial community. So the dots show the abundance of particular bacterial strains in a particular genotype. So you see that there were differentially abundant taxa according to the particular host genotype. So the conclusion is actually, I'm going too fast. The conclusion is that both microbial inoculum affects what you get in the gut microbiome, and also the host genotype plays a role. And these are a bit correlations based on the same experiment, but also on other experiments. But what is also interesting is that the more diverse your microbial inoculum is, so the more bacterial strains they are exposed to, the more diverse the gut microbiome will be. So the more strains you give, the more they will take. So that's relatively easy. But then from another experiment, we actually also saw that that affects the Daphnia performance. So the richer the inoculum is, so what you see on the x-axis, the OTU richness is actually the richness of your inoculum. Then you also see that this has a positive effect on the growth of the Daphnia. So the more diverse the inoculum was, the better the Daphnia could grow. So there's clearly also a positive effect of a diverse community on the performance. Okay, so these were a few general aspects from Daphnia microbiome interactions, so more focusing on microbial genotype interactions. And like I said, the one system we are particularly interested in are these toxic cyanobacteria. So these are toxic bacteria. And so Alice, Maxime, and Emma did an experiment, and where we actually compared different, so these are field strains, so field genotypes. So we compared different Daphnia field genotypes and exposed them to different cyanobacterial strains. And the question in here is if you combine contemporal associations, so late Daphnia with late cyanobacteria versus early Daphnia with early bacteria, and you do that for 2 years, if that has an effect on how well the Daphnia deals with the cyanobacteria. And that's actually what the results show. So the results show that, so the most important effect that you see here, so is the contemporality. So we see that there was an effect of contemporality on the survival of Daphnia. So if they got cyanobacteria from the same time window, they survived better than if they got cyanobacteria from a different time window. And typically this was also characterized by genotype-genotype interaction. So different genotypes were responding to different cyanobacteria genotypes in a different way. So clearly this reflects dynamic interactions between these Daphnia genotypes and the cyanobacteria genotypes. And then the question is, do the microbiota play a role in that? So our hypothesis was, okay, we see these genotype-genotype interactions. We know already there are genotype microbiome interactions. So the next question is if there are genotype microbiome genotype interactions. And so these are experiments Emily did a few years ago in my lab. So to assume that there is genotype microbiome interactions, so you actually assume that your tolerant genotypes and your susceptible genotypes have different microbiomes. So that's the first assumption. So your tolerant phenotypes would have a different microbial community than your susceptible phenotype. And then the second thing that we questioned is if you can also modify your microbial community by exposing your Daphnia gut microbiota towards the toxic cyanobacteria and see if you can modify the community and in that way actually modify the phenotype of your Daphnia. So that's a big transplant experiment Emily did. So what we had is the top panel is the donor phase. So in the donor phase, we exposed different genotypes. So the red ones are the susceptible genotypes. The green ones are the tolerant genotypes. We exposed them to actually a control diet, a control algal diet, or a cyanobacterial diet. So that was actually microbial evolution of your microbial community towards toxic algae versus not. And then we took out the microbial inocula and transplanted them in the same genotypes that were made sterile, so the bottom panel, and looked if we can modify the phenotype. So simply said, if you can make a susceptible genotype more tolerant by giving it the right microbial inoculum. So, and if you can modify that phenotype, that's actually a proof for this genotype microbiome infections. So that's actually also what Emily saw. So so there was clear an effect of the host genotype in the microbial inoculum. So if the recipients got microbiota from a donor genotype that was tolerant, the recipients were also doing better than if they got a microbiome from a genotype that was susceptible, first of all. And then secondly, we also saw that the diet the donors had was also reflected in the recipients. So the graphs on the right are the survival graphs of the recipients. So you see that if the recipients got microbial inocula that have seen cyanobacteria before, the survival will be better than if they didn't get that. Okay. So that actually shows that we have this genotype microbiome interactions. We could play an important role in modifying these environmental responses. But then a second thing that we actually also saw in our recipients, so like I said, we had an effect of the microbial inoculum on the recipients. The thing is, in the beginning, we didn't see a recipient genotype effect. I wasn't talking about recipient genotypes. I only talked about the donor genotypes. But that actually shows that in the beginning, the strongest effect that you have is a more inoculum effect. So, and that we investigated by a follow-up experiment. So that was a more follow-up experiment on the previous experiment where we looked to 2 different genotypes only and exposed them to 4 different microbial inocula. And if you look there you see in the beginning, the strongest effect on the microbial community was explained by the inoculum. So the community that you got in your Daphnia was mainly reflecting what they got, the inoculum. But when you let your Daphnia and your microbial inocula interact, the recipient genotype became important too. So that actually reflects that there's a kind of interaction between your host genotype and your microbial inoculum occurring over time. So there's a time-dependent effect. And that's actually also important because if we would have had the genotype effect immediately from the beginning, it wouldn't reflect our genotype microbial interactions so strongly. Okay, then the second thing, so that was more transplant experiments from the lab. So the other thing we did, that was in a framework of a bigger study. So it was a study where we compared Daphnia and microbial inocula from different ponds over Flanders. So, and it was initiated by Lien and Lore, who were involved in that big project. It was also related to urbanization, seeing the effect of urbanization on different microbial communities. But actually, what we did there was doing transplants from microbiota with different genotypes, and there Shira was involved to look if there, first of all, if we see local adaptive patterns, and if these local adaptive patterns are mediated by the microbiome. So what Shira actually did, so the K is for City Kortrijk, that's where my interdisciplinary facility is, where my research group is hosted, and the L is for Leuven, where the main campus of the university is. So what we did is taking ponds from Kortrijk and ponds from Leuven, and then we made the different associations. So this time it's not contemporary, but it's sympatric. So it's comparing associations, Kortrijk sympatric, Leuven sympatric, or allopatric, Kortrijk Leuven Kortrijk, and then expose those combinations again to the toxic cyanobacteria and see if there's an effect of where the microbiome was coming from. So what we transplanted were the microbiota, and then we again did the survival experiments. So what Shira actually saw, and there's a bit of variation between the different clones, so the letters that you see are the different clones, but if you focus on the full lines, so the full lines are the lines from which the donor microbiome has been exposed to the toxic cyanobacteria, versus the dotted lines didn't have exposure to the cyanobacteria. And the difference is then if there are sympatric microbiomes or allopatric microbiomes inoculated to the Daphnia. and then re-exposed to the toxic cyanobacteria. So if you do that, you can actually see that for most clones, not for all, but for most clones, the sympatric association had a higher survival than the allopathic association. And then, very interestingly, if we look to the microbial communities on the right, so what we see in the sympatric associations, we saw less variation in the microbial community in comparison with the allopathic associations. So if you look to the microbial community of the allopathic associations, there was actually clearly no trend in the community that you found. But if you look to sympatric associations, it looked like the Daphnia seemed somehow to select always for the same microbial community. And I must say that we only used one cyanobacterial strain in the final exposure here. So it was all towards the same direction. So that seems to show that actually this microbiomes may also play a role in the local adaptive responses. Anyhow, that if they get the microbiome from the same pond, they are somehow better protected towards stress than if they get microbiota from another pond. So, okay, what I more or less showed you so far is the top panel. So showing that the environment or the diet has an effect on your microbiome, and that this also modifies your phenotype. And we also see that the Daphnia genotype has an important effect on the Daphnia phenotype. But what I didn't show you yet is the bottom-up feedback. And as I said in the beginning, to have this hierarchical eco-evo dynamics, we actually also should have this bottom-up dynamics. So we should also see that if you have a modified phenotype, that you can also somehow structure your environment. And if that happens, you have actually at least potential for this eco-evo dynamics to occur. And therefore, we did some additional experiments, which were also done when we did all these transplants. So it was also Emily who did that. So we again compared different genotypes, but then now more than 4. So it was 9 genotypes, and exposed them to control diet versus a cyanobacterial diet. And then we also looked actually if we also found an effect on the bacterioplankton community. So not only on the gut microbial community, but also on the bacterial community. So if we take donors and we integrate it in transplants, do we also see an effect on the bacterioplankton community? And if we look to those results, well, first of all, what we see is that in general, the bacterioplankton community is different than the gut microbial community. So there seems to be a separation. So what the Daphnia has in the gut is different than what is in the environment, but there is a small overlap. But then very interestingly, so the gut, again, so this is the results from the gut microbiome on the right. So the C panel is the microbial community from the gut microbiota. There again, we saw the genotype effect. So which genotype got a particular inoculum also was reflected in a different microbial community. But also in the bacterioplankton, the genotype effect is also there. It's much less strong because the diet effect is stronger than the genotype effect in the surrounding bacterioplankton community, so on the B panel, but it was definitely there. So we did some additional network analysis to really disentangle what had an effect on what. So that was quite complicated analysis. So Francois Massol helped us there. And then, yeah, these are the type of analysis we did, but it was mostly via RDA analysis. But if you look to those analysis, so the left panel is actually if you compare your communities together, so the bacterioplankton and the gut microbial community in one analysis, that you clearly see again, there was an effect of the type of community, which we saw. The gut was different from the bacterioplankton community, and also the diet had an effect on your community. But when you then looked more in detail to the sub-networks, so if you look to the gut microbial community only versus the bacterioplankton community only, we also saw a signal of the host genotype on that bacterioplankton community. So in here, we got actually an extra confirmation that host genotype also played a role in the bacterioplankton community. So that somehow shows that depending on which host genotype you have in that whole interacting system, that also the final outcome is different. Yeah, so we look to cyanobacteria. We also look to other stressors, and that's a bit, so especially, so on the right, you see here again, these are the cyanobacteria. So what we do there is looking more to strain-strain interactions. So the interaction from cyanobacteria on infections. So this is a viral infection, so it's white fat cell disease. What we already noticed before is actually that this cyanobacteria also affect infections in Daphnia. So if you have particular strains present, the outcome of your infection may differ. So what we saw is that there was a kind of protective effect of particular cyanobacteria strains on infection outcome in Daphnia. Yeah, we looked also to hypoxia, but I won't tell too much about that. So that's research I did already. It's compilation of different papers where we actually see these strain interactions, which are actually also microbiota interactions in Daphnia. So also for particular, more symbiotic, parasitic symbiotic. So for instance, some parasitic microsporidians in the gut, if you have them, they were also like giving a more protective effect on, for instance, this white fat cell disease. So we have seen that before, that the presence of a particular symbiont affected the virulence outcome of other infections. So that shows that there are different strain-strain interactions. So you have this whole microbial pool of interactions in such a simple host as Daphnia, and that seems to mediate the responses. And that's another experiment Shira and Laura did, and it's almost, how much time? I still have some time. But so this is an experiment where we exposed, again, 3 different clones, and then we exposed them to a lab microbiome and a natural microbiome, and we did different stressor treatments. So the stressor treatments we looked at here were controlled, so that's not stress, but then here an omicete infection, and then the toxic cyanobacteria, and then the combination of the two. And what we noticed is that when you give them a lab microbiome, you actually see the antagonistic interactions between these two stressors. So there was actually a more tolerating effect of the cyanobacteria on the infection. So in total, the infection was less virulent when the cyanobacteria were present. But when you give them a natural microbial inoculum, you don't see the effect. So if you give them a more complicated microbial inoculum, the antagonistic interactions between the 2 different stressors disappears. So that shows that well, clearly this microbiome plays a role, and it's more complicated as we all thought originally. So we do see different interactions. Yeah, just that is just to show you that we're also doing this with temperature adaptation and pesticide adaptation, different experiments where we expose Daphnia towards different temperatures and pesticides, and see if the microbiome plays a role. But in general, actually from these transplants, we can conclude that both the bacterioplankton or the microbial inoculum and the Daphnia host genotype shape the Daphnia gut microbiome. So we get a kind of selective interaction there. And we also see that Daphnia gut microbiome drives adaptation and acclimatization to stresses, and that there's definitely a scope for the microbiome to shape this eco-evolutionary dynamics, which affects host community and ecosystem dynamics. So this hierarchical eco-evo dynamics. Yeah, we do this also, so that is a bit a spin-off project, so not looking to cyanobacteria, but then to microplastics. So a lot of people are working on microplastics this time. So we were actually also playing with the idea. So we know that particular bacteria can degrade particular plastics, microplastics. So the idea is here, if we expose Daphnia with their microbiome towards different microplastics, do we see shifts in community, and are these shifts in community associated with different functional responses? So we do exposure experiments, and it's also like in the framework of a big field study where we compared ponds with high plastic presence versus low plastic presence, and then compared the different microbial profiles. And actually we see that in the ponds where there's a lot of plastics presence that there's in the functional profiles of the microbiomes, you also get more plastic degradation associated genes. So that's only done on PET because that's the only plastic where those degradation pathways are known from. So then you can look into the data. But this is a collaboration I'm doing with Caroline Faust on the right, who knows much more about bioinformatics than me. So they're helping a lot in there. But it's a combination of experimental work and field work, trying to see if the microbiome also can play a role in that. Yeah, this is a bit another project where we also look to different, it's not Daphnia, it's on Hamarus, where we see in different environments if we characterize the microbiomes and link it to particular infections in the Hamarus, if we see responses. So if stressed environments do have different microbiomes. And just one of my final slides, so I'm moving a little bit away from the Daphnia, other projects, so there are also people where I'm collaborating with. So on the top left is the project of Ruben. So that's also an aquatic project. It's where we look to the snail microbiomes to see if we can find particular bacteria that deliver tolerance or resistance towards schistosome infections. And that's put in, it's more the framework of the One Health framework to find if you can find this particular bacteria. And then secondly, that's one of my wildest projects on lemurs with Karine in Madagascar, where we also look disturbed, undisturbed areas for microbiome community differences. And then a few other spin-off projects. The wildest one is definitely also this human microbiome project, where we actually want to link human genetics towards microbial community profiles to find if we find a pattern there. Voilà, this is the institute where I work, so always welcome to visit if you want to see that. So we have experimental facilities. We also have outdoor areas where we can manipulate. This is the team working on particular aspects that I have shown. As you can see, I'm collaborating with a lot of people, so I'm really into the team science rather than individually. So thank you very much for your attention, and I'm open to questions if you have some.